All right, Josh, I'd say let's go ahead and start.
Okay. Okay, we'll get started here. Okay, can you all see that?
Yes.
Mm-hmm.
Okay, great.
You have
So,
you have
good.
78 participants before you switch meetings. So there's 25 left to sign in.
so I'll take it slow at first, and then if they get here, they can kind of realize we got 54 now. We are getting people coming. You want me to start? I'll kind of go slow at first, and then move faster as I go through this. Is that okay?
Please start.
Okay.
Yes.
Okay. So, hi everyone. I want to thank everyone for for coming to see my talk here tonight. I want to thank JP Cavagelli for having me give this talk. The talk I'm giving to you tonight is actually about the ecological role of ammonites in the late Cretaceous seas of North America, and it's based on research that I've been doing and also numerous researchers actually all across the globe. So what I'm going to be sharing with you is a lot of the recent stuff we've figured out about ammonite paleobiology over the last 20 years, and specifically focusing in on the late Cretaceous. So the last like 20 million years of the Cretaceous before the ammonites actually went extinct. So that's going to be the focus of the talk today. Let's see here. Going. Okay, there we go. Okay, figure out this guy. So just to start off, if you haven't ever seen an ammonite before, I suspect most people here are familiar with ammonites, but ammonites are a type of cephalopod. They're actually named after the god Ammon right here, which was an Egyptian god of the sun. So you can see he had these ram's horns on the side of his head that look pretty similar to an ammonite. And so when they first named ammonites, they thought it looked a lot like the god Ammon, and they applied the name there. There's ammonites, just like other cephalopods, likely had like arms and tentacles. These are reconstructions of different ammonites below. Can you see my mouse there? Is that something people can see? Anyone?
No. Yes.
Yes.
Okay, okay, good. I want to make sure you're seeing things. So I put a couple of reconstructions of ammonites from the late Cretaceous on here. So right here you have a reconstruction of Placenticeratidae, and right here you have a reconstruction of Scaphitid. Most people, when they draw or reconstruct ammonites, they usually they reconstruct them with just like a squid in the shell. And ammonites were not just squids in shells. They were very distinct organisms which had evolutionary relationships with squid, but also had their own unique anatomy, just like all modern cephalopods do. So this reconstruction right here of a Scaphitid is probably one of the most accurate ones that we've actually done in the last that have come about actually. So this was put together by Neil Lamman, who specializes in Scaphitid and other Scaphitids. And this is probably one of the most accurate ones. And you can see right here how they looked. So they had they probably had 10 arms, so similar to coleoids. And they had this small, probably a hectocotylus, which is their reproductive organ right here. At least the males did. So this arm right here would actually break off and carry sperm with it and fertilize the females. And most ammonoids, I'll go more into detail about that, have very distinct mouth orientations. So, and another thing about ammonoids is compared to other invertebrates, they also get very large. And you can see from this picture right here, this is actually in the Smithsonian, but you can see this large pachydiscus here. This ammonite got up to like 3 feet in diameter. There's also other large ammonites. The largest ammonite actually that ever lived during the Cretaceous, and it probably got up to 3 meters in diameter, so like 9 feet or so. So they got very large. So to give you a little more background about ammonoids, they look very similar to the modern nautilus, at least the shell shape does. So they're ectocochleate cephalopods, which means they had an external shell. This is in contrast to things like the coleoids, which are the octopods, squid, and which are living today, and the vampire squids, which have usually internal shells. So they have very similar shells to the nautiloids, but they're not as closely related. And I'll talk about that here in a second. One of the primary differences that actually defines the ammonoids is their septa. And I suspect most people, if they've seen ammonites before, they have these very distinct chambered shells, just like the modern nautilus. So in the modern nautilus, they use these chambered shells for buoyancy. So inside of each of these chambers, they grow these chambers throughout their lifespan. And these chambers are interconnected with a vascularized tube, which actually exchange fluid and gases through osmosis, and then regulates the buoyancy of the nautiloid in the water column. Ammonites probably had a similar process. So like this right here, you can see they have these, right here you can see the septa of the nautiloid, and they're very simple shaped. In contrast, ammonoids have these very complex septa, which produce these complex suture lines, which is actually the defining characteristic of ammonoids, these very complex subdivision of their chambered shells. Now, why they had more complex septa compared to like modern nautilus is probably related to their rate of growth. Ammonites probably grow much faster compared to modern nautilus. So modern nautilus take around 30 years to grow, and they have very thick shells. And the thickness of the shells and the simplicity of their septa is probably related to inhabiting much more deeper water column, and also having a greater, investing more energy into shell growth. Ammonoids, in contrast, had much thinner shells and probably had to strengthen those shells because they were much more thinner. So they developed this much more complex, septal pattern to strengthen the overall shell. So these septal patterns, a lot of people think that they're used for identifying the specific species, but they're really only good to probably a genus. There's a lot of other characters that, as an ammonite expert, I prefer to use, usually related to the external morphology. But you can see right here how complex they are. So when we think about ammonoids, people usually try to relate them either to the modern coleoids, like the octopod, squid, and vampire squids, or they compare them to modern nautilus. So the question is actually, what are they actually most closely related to? And originally back in the 1800s, early 20th century, most people thought because they had external shells that they were very closely related to the modern nautilus. They even made a lot of early reconstructions similar to modern nautilus when trying to reconstruct these. But over the last few decades, we've started reinterpreting actually how ammonites are related to other cephalopods, and we now think they're not so closely related to the modern nautilus. They're probably more closely related to the coleoids. So the reason we think they're actually more closely related to squids and octopods is related to their how they reproduced. So ammonites are, so if you look at modern cephalopods, the nautilus usually lives around 30 years. It only produces a small number of young when it reproduces. It reproduces multiple times. When it reproduces, also lays probably around 30 eggs total. And it puts a lot of energy investment into those eggs. So those eggs have large yolks. And the eggs, when they hatch, are around, this is actually a hatchling right here, around 2.5 centimeters or about an inch. Modern octopods and squid, they actually invest in large numbers of small hatchlings, so small juveniles. So they produce millions of eggs. You can see that right here with this modern octopod with all the egg cases right here. They have thousands and thousands of eggs, sometimes 10 to 100,000 eggs. And all the eggs are very, very small. So they don't put a lot of investment in there. And it's probably also related to how they well, what's preying upon them in the long term. Ammonoids, in comparison to the nautilus and the colloids, are much more similar to modern colloids. So they produce thousands of young, small eggs. And you can see that right here. These are actually ammonite eggs from the UK that were published on just, I think, around a decade ago. So these were the first ammonoid eggs ever described, but they're all very tiny. So you can see this scale right here. It's around 1 centimeter in size. The eggs of ammonites are very small, just like modern colloids. So, and when you find the hatchlings, they're pretty common actually in the fossil record. They're usually very tiny. So we think most ammonoids were probably produced masses of eggs, and then they were all very tiny, and they didn't have very much yolk. They probably hatched within a few weeks after being laid. And they kind of had, the best way to describe ammonoids is they had a live fast, die young kind of strategy. So based on isotopic analyses, most ammonoids, we think, probably only lived around 2 to 3 years. And that's pretty similar to the life strategy of modern colloids. In comparison, like the nautilus that lives today lives like 30 to 40 years. So much different life strategy. So you can see this right here. This is a reconstruction of the phylogeny of modern, fossil and modern cephalopods. So there's the cephalopod stem groups, and those are things that are probably lived during the Cambrian. They're these weird little limpet-like organisms that we think are like early cephalopods. Then there's the Nautiloidea, which is probably the least derived of all the cephalopods. And then in between the nautiloids and the coleoids, these are the coleoids right here, which include the decapodoforms, which are things like squid, and then the octopodoforms, which are the octopods, and the Belemnitida, which are the belemnites, these kind of ancient squid ancestors. They're all within the coleoides. So they're actually more closely related ammonoids to the coleoids than they are to the nautiloids. And just they have much more derived features, despite retaining the ectocochleate shell. So ammonoids are really important for biostratigraphy. I mean, just absolutely amazingly practical. They've been used all across the globe to actually subdivide rocks ranging from the Devonian all the way to the, well, the end of the Cretaceous. So just to give you an example of how useful they are, is they actually evolve ammonoids just in general at a much higher evolutionary rate as compared to other like useful biostratigraphic indicators like foraminifera. Foraminifera usually have a range of around 1 million years to 500,000 years. Ammonoids usually evolved at a rate of around 250, 500,000 years. So ammonoids are much more useful for actually subdividing rocks. And you can see that with this chart right here. So this chart, what I'm showing you right here, is a biostratigraphic chart developed by William A. Cobbin, who passed away probably around 10 years ago. But William A. Cobbin was the foremost expert on ammonites of the Cretaceous of North America. And he spent his entire career, all the way from the 40s to the 2000s, reconstructing the biostratigraphy and also describing the ammonite faunas of North America. Through his work, this is one of his last bits of work that was published in 2006, but he defined 67 ammonite zones over the late Cretaceous or upper Cretaceous. And within those zones, each zone represents probably a time interval of around 250,000 years to 400,000 years. Some are longer, some are a little bit shorter. So this work kind of grew over time, and it's an amazing piece of work. And it's probably actually the highest resolution record that you can find actually globally for deep time. You can also find similar kind of biostratigraphic subdivisions also for the Jurassic of Europe, which are amazing as well. But for the Cretaceous, probably the best record here is in North America. And the common ammonites that we actually find here in North America are like these baculites, and we use for biostratigraphy are these things called baculites, these things called Didymoceras, and then the Scaphitids. So they're very practical for actually subdividing and correlating rocks. So today, what I'm going to focus on is actually just the last probably around 20 million years. So mostly the Campanian and Maastrichtian. So these intervals of the Cretaceous. Right here This is the very end Cretaceous at 66 million years ago. And I'm going to go back to around 83 million years ago and talk about the dominant Ammonite faunas that occurred in North America during that time and what their ecology was. So during that interval, during the Campanian, the North America was actually flooded by what's referred to as epuric seas. These are shallow seas that flood the interiors of continents. There were also neritic seas, which flooded the margins of continents. So you can see that right here. We had a major epuric sea called the Western Interior Seaway that stretched from the Arctic Ocean all the way down to the Gulf of Mexico. And the seaway was also interconnected with the Atlantic through the Hudson Seaway. And over time, the seaway actually shrunk from the Campanian into the Maastrichtian due to the growth of the Rocky Mountains. So starting around 77 million years ago. So you can see that with these two pictures right here. The shoreline and the size of the Western Interior substantially shrunk. And right here, this is the Maastrichtian right here. This is the time interval where you have things like dinosaurs like T-Rex and Triceratops. And those kind of animals were living on these shallow coastal environments adjacent to the Western Interior Seaway. So Ammonites are also found abundantly in the Gulf Coast and the Atlantic coastal plain. They have not been as well described, so as compared to the Western Interior. So they're still being worked on, especially their biostratigraphy and also their the diversity of the faunas. The diversity actually outside the Western Interior Seaway is actually much higher. So it's much more diverse, but the abundances are within certain intervals are very rich and other intervals are much lower. In the Western Interior Seaway, they're almost always super abundant, but they have low diversity. So let's start off and just ask, so where did Ammonites live and what did they eat? Because that's really the focus of this talk. What did they do for their life habit? So when we're trying to interpret the ecology of Ammonites, we usually try to look at them in the context of modern cephalopods to see how they compare. So modern cephalopods, we have different actual habitats that they live in. And we think Ammonoids probably also lived within these similar types of habitats. So today there are nectic cephalopods, vertical migrants, and nectobenthic. Nectic is cephalopods that live up in the water column. And then nectobenthic refers to Ammonites that live close to the sea bottom. And then they don't live actually on the sea bottom, they live close to it. And then the vertical migrants actually probably migrate up and down through the water column. So most modern cephalopods like squid today are probably nectic. They live actually in the water column, whereas octopods are typically nectobenthic. They live close to the sea bottom. There are some exceptions to this, like the Argonauts, but most octopods live close to the sea bottom. Nautilus is a vertical migrant, so it actually moves up and down. And it's kind of cool. In the last few years, there was an exciting paper published by Ben Linsmeyer that actually shows that you can see the migration habits movement in the stable isotope geochemistry preserved in the shell of modern Nautilus. This has yet to be shown in Ammonites, but there's some suggestion that some Ammonites might have actually been vertical migrants. So we probably have all these different types of habitats reflected in Ammonites. Feeding hypotheses. Some people have suggested that, if you look at modern cephalopods, there are macrocarnivores like octopods, there are scavengers like Nautilus, and then there's also the microcarnivores like the vampire squid. So these are things that passively float in the water column and eat small plankton. And we think Ammonoids probably had all these variable niches as well for feeding. So, but how do we interpret the life habit, the feeding ecology and the habitat of Ammonoids from the fossil record? Because typically all you find are just fossil shells. So there are several ways that we can actually do that. One thing is actually looking at the associated fauna. So what kind of Ammonites co-occur like with like what organisms co-occur with them. For example, if you find ammonoids that are mixed in with like diverse mollusks like gastropods and bivalves, then you could suggest that they probably could live actually in fairly like, diverse communities. But if you find ammonoids that are living in facies which are devoid of all benthic faunas, it would suggest that they're probably living high up in the water column, probably above a disox or a noxic facies. And we can support that kind of like data with looking at the lithofacies. So look at what types of rock record they're being preserved in, if they're being preserved in like organic carbon-rich facies versus, let's say, sandy, shallow water facies. We can kind of figure out where they live based upon what kind of facies they lived in. Traditionally, people didn't think that was actually very practical, and people have argued against that because ammonoids have these like hollow shells. So this is a model right here. I don't know if you can see that very well, but they have these chambered shells, and after they would die, the ammonoids, some people have suggested that ammonoids would float around after they die and be deposited in different environments that were not reflective of their original habitat. But over the last few decades, people have actually started doing analyses of modern nautilus, and they've actually shown that's actually pretty unlikely to occur. And they've also done modeling with ammonoids, and it shows that ammonoids probably did not float around much after they died. So they probably, most ammonite occurrences probably occur close to where they actually lived. So when we find a fossil, they weren't floating around great distances. Some probably did. Ones that, specifically ones that looked a lot like modern nautilus, had a more likely chance of actually passively floating around. But for most ammonoids, they probably didn't float very far. They probably sunk within a few hours after they died. We can also use the shell geochemistry. So that's like stable isotopes. So what we can do is the shell geochemistry, ammonites are kind of unique. They actually take in the seawater and incorporate the chemistry of the seawater into their shells. And so the signal of where they were living in the water column, which changes due to depth, is reflected in their shells. And what we can do is actually compare the isotopic, like geochemical values of ammonite shells to the isotopic values of gastropods and bivalves living at the sea bottom, and compare that also to the geochemistry of shells of little microscopic organisms called foraminifera living high up in the water column, and kind of figure out if ammonoids were living high up in the water column or near the sea bottom. And so it kind of gives us some details about them. We can also use their shell morphology, which is just like the shape of their shell. So we can look at if they were living in if they were kind of compressed, more active swimmers versus, let's say, something that was more, like a passive floater like this ammonite, Didymoceras, that wouldn't be very much of an active swimmer. We can also look at the orientation of their aperture. The aperture is the opening where the animal's head sticks out. And so like this right here is Didymoceras, and its aperture actually likely pointed upwards. So we can look at the orientation of the shell and how it floated in the water column and figure out if it pointed upwards or downwards or different directions. And that can give us some indication of where these ammonoids live. We can also, one of the most exciting things is actually looking at the jaw structure. So ammonoids have these very distinct jaws, and which are, all cephalopods have these jaws that are almost beak-like. And based upon the thickness and the structure of the jaws, we can start to figure out what type of food the ammonoid was living or eating, consuming when it lived. We can also look at the radular structure. This is something that's been only done in the last 10 years. So right here, these are the radular structures of ammonoids. So this right here is the jaw structure, and this is the radular structure. So here's the jaw, and right up above that is the radula. And we can look at the radula and compare that to modern coleoids and nautiloids and see how similar that is to those groups, to see if they were consuming different types of prey. So, and it appears actually based on the few studies that have been done so up to this date, is that they actually compare very favorably to certain like cephalopods living today. So their jaw structure at least, which tells us they converged on similar feeding ecologies. And there's also the remnants of the last meal. And this is debated if they actually are the remnants of the last meal, but if you look at this right here, this jaw, this is the radula, the orange part right down here. And above this, they think this is the gut content. They found this with other fossil cephalopods. And in this gut content, they found pelagic octaval gastropods and also tiny little crustaceans. So we can kind of use that information to figure out what they were eating.
Joshua,
Yeah.
I'm so sorry to interrupt, but we just got a 10-minute warning.
Oh, we got a 10-minute warning? Oh boy. Okay.
We're down to 6 minutes and 23 seconds.
Do you want to?
8 minutes, 22.
Oh, okay. So I'll try to get through this.
We can set up another meeting. I could do another meeting in that amount of time, so we can have another 40 minutes.
Okay.
Kelly, you're on.
You want to do it?
Let's do it.
Okay, let me figure.
Thanks.
You want to figure it out and I keep talking?
Yeah.
Keep talking.
Okay. One of the other most exciting thing that's actually been happening is there's actually the next speaker for the Tate's lecture series is going to be David Peterman. And he's been actually doing some of the most exciting work out there. He's been creating these models. So I got these actually from him. And he's been figuring out how ammonites actually swim and orient themselves actually in the water column, making these models. And he's even making a robotic ammonite. So real exciting work. And if next month, if you have a chance, watch his talk, because that's some of the most exciting work on ammonites being done. So this is how we figure out the ecology of ammonites. So let's talk about kind of how we group ammonoids, like phylogenetically.
So
What?
let me interrupt one more time. If this meeting ends, I set up another one with the exact same ID and passcode. So if it cuts us off, just rejoin with the same one.
Okay. Sounds good. Okay.
Resend the ID and the passcode, please.
I'll put it in the chat.
Okay. Okay, so traditionally, historically, people grouped ammonoids based upon their overall shell morphology. That was the traditional way. So they based them on either if they were aberrant shell forms or planospiral shell forms. So planospiral are what you typically think of as ammonites. They have normal coiling to them. And then the aberrant forms were ones where the coiling actually detached and separated, like this Didymoceras right here. So you can see that this model right here, where they detach the coiling. So most people like grouped them based upon their shell forms historically. And what we've actually found out over the past few years is that this probably doesn't actually mean much. And what they've actually started finding out actually is that their jaw structure probably is more significant for grouping ammonoids, so evolutionarily. So today, what we do is we break ammonites into Aptychus type jaw-bearing ammonites and Rhynchaptychus type jaw-bearing ammonites. So the Aptychus type jaw-bearing ammonites are, I like to call them the taco-jawed ammonoids. And they have these very fragile like jaws, which look almost like a taco. So it's almost like a taco shell like that. And very fragile. The Rhynchaptychus type jaw-bearing ammonites are these ones that have very prominent beaks that are very robust and very sturdy, very similar to modern nautilus. So within the Rhynchaptychus type jaw-bearing ammonites, we actually include Litoceratina and Phyloceratina. And then in the Aptychus type jaw-bearing ammonites, there's Ancyloceratina and Ammonitina. We think that Ancyloceratina and Ammonitina probably evolved from Litoceratina. So during like, I think the end Triassic, early Jurassic time. So the Rhynchaptychus jaw-bearing ammonites, they're usually what we think of as probably macrocarnivores or scavengers. And that relates to their very thick, prominent beak-like jaws that are similar to nautilus and also modern octopods. The Ptychophora So that's the group that includes the Ammonitina and the Ancyloceratina. They have those taco jaws, which are very fragile, just like a taco shell. They were probably planktivores or gelatinivores, so what we think of. So they were probably eating things like jellyfish or probably small zooplankton, so small crustaceans, small gastropods, and probably had an ecology very similar to modern vampire squids. Now, the unique thing about these ammonites is that they lived in shallow water environments. Today, there are no cephalopods with this type of ecology living in shallow water. Most of them lived in deep water. So, in today's talk, what I'm going to focus on are the ammonites that are most abundant in North America during the late Cretaceous. So I'm only going to focus on the Ammonitina and Ancyloceratina. So the Ammonitina are the normally coiled ammonoids, and the Ancyloceratina are the heteromorphs or the aberrant forms. These are the ones that have detached coils. So Ancyloceratina are actually the most abundant ammonoids that we have in North America, and they're also the ones that we use primarily for biostratigraphy. Ammonitina are very common, but not as abundant as these guys. And they probably, each different group probably had their own distinct ecology. So let's start off with the ecology of Ammonitina. So, and the reason, real quickly, just to mention why I'm not mentioning the Litoceratina and the Phylogeny is because these taxa are usually deep water ammonites. So these are ammonites that live in deeper species, probably more oceanic settings. In North America, we didn't have a lot of oceanic settings preserved in the rock record. So most of our rocks that we find are usually shallow water facies, like in the Western Interior. And shallow water facies are dominated by the Ancyloceratina and Ammonitina. So let's go on and talk about Ammonitina, the planospiral ammonoids. So the first group I'm going to talk about are the Placenticeratidae. So Placenticeratidae are the most abundant planospiral ammonites that we find in the last 20 million years. They actually went extinct probably around 6-7 million years before the end of the Cretaceous. We have a lot of data on them and a lot of information about their paleoecology. So what we know about them is that they were likely nectic macrocarnivores and that probably inhabited the surface waters. And the way we know that is based on their stable isotope geochemistry and their facies distribution. They can be found in all different types of facies, including anoxic facies and also well-oxygenated facies. So they probably were living well above like any kind of dysoxic environments. And they could also live in well-oxygenated facies. Also, their stable isotopes are always unique. So they're distinct from organisms that live on the sea bottom. Their stable isotopes seem to reflect organisms that live more higher in the water column. Now, their jaw structure is interesting. It's more prominent compared to the Ancyloceratina, and it's more of a, it's not as, it's more beak-like, but not as sturdy as like a modern nautilus or octopod. So what people think is that they were probably munching away at like gelatinous zooplankton, so like jellyfish, and possibly squid and small fish. And you can see a picture right here of Placenticerus, what we think they possibly look like. I actually don't think this is actually a good reconstruction. There hasn't really been one to date, actually, of what they likely look like. It's hard to interpret them. There's also the Sphenodiscids and pachydiscids. Okay, so let's continue on. Okay, I was talking about the planospiral ammonoids, Placenticeratidae, and I started moving on into the other planospiral Ammonitina, which are the Sphenodiscids and pachydiscids. These are also very common planospiral ammonites in North America during the late Cretaceous. We don't know much about their paleoecology compared to the Placenticerids. So they look almost exactly the same or very similar in morphology, and so like the Sphenodiscids seem to come in after Placenticerids went extinct. Pachydiscids seem to persist throughout the entire time and preserve, like prefer deeper water environments. So they're common in the Gulf Coast, even up to the late Cretaceous. They're probably one of the most abundant ammonites you can actually find in certain deeper water facies. We don't know much about these guys, and they haven't really been as well studied. There's a recent paper that's eventually going to come out here soon about the Sphenodiscids stable isotopes. These guys, the Sphenodiscids, seem to prefer shallow water facies, usually in the near shore. The pachydiscids seem to have lived up in the water column and were probably living more offshore. They did not like going into these shallow nearshore facies. Both are probably nectic macrocarnivores like the Placenticeratids, but we don't really know. So there hasn't really been any description of their jaw structure or what they were feeding on or any information about that. So we really aren't sure what they were doing, but we think they probably had similar ecologies based upon their shell morphology. When we look at the Ammonitina, these planospiral ammonites like Placenticerus, Sphenodiscus, and Pachydiscus, we think and this is actually suggested by Neil Lamben, is that ecologically, they're very similar to the modern sunfish. So if you look at this is a picture of the modern sunfish, these things get absolutely immense. And these Ammonitina had also got very large and very compressed. So sunfish are kind of a compressed fish that eats gelatinous organisms, so like jellyfish, small fish, small things, usually small vertebrates, squid. And they look very similar to the Placenticerus. So we think because of what we can find out from the jaw structure of the isotopes, that they had the similar ecology to modern sunfish. So sunfish probably have a similar role ecologically to the Ammonitina. So let's talk a little bit about the aberrant ammonoids or Ancyloceratina. So these are what people typically call heteromorph ammonites. Heteromorph doesn't actually mean much phylogenetically. It's just kind of an outdated term. The better term to use is Ancyloceridids or Ancyloceratina. So I'm going to go through the most abundant Ancyloceridids. The first one I'm actually going to talk about are the most common that you find in North America. And those are the Baculitidae or the Baculitids. So the Baculitids are these straight-shelled ammonoids. And if you were to go to any Cretaceous rocks from the California coast to the Gulf Coast to the Atlantic coastal plain into the Western interior, you're likely, if you're going to find an ammonite, it's likely going to be a Baculitidae. So usually Baculites specifically. And we have actually more information on the ecology of Baculites than any other heteromorphic or the Ancyloceratid ammonoid. What we know about them is actually they probably were vertical migrants. There have been actually conflicting stable isotope studies on their shell geochemistry. Some people actually, a couple recent studies have suggested they live close to the sea bottom, while older studies or studies looking at larger individuals have actually suggested that they live actually higher in the water column. And the facies data suggests they probably lived higher in the water column or were probably vertical migrants. And I think some new data that hasn't really actually came out yet is that they were probably moving up and down in the water column. That kind of research will be published in the next few years. So we think they're probably vertical migrants. We have their jaws and their radula actually preserved, and also the remnants of their last meals. And their jaws are actually probably the most fragile and thinnest jaws of all the ammonoids, and which suggests they weren't eating very large prey. They were eating small things. Their radula structure resembles the radula structure of deep sea squids and vampire squids, which suggests they were eating probably plankton. And also they found inside this interpreted gut content, they found actually pelagic gastropods and small crustaceans. And also their orientation of their shells. So here's a good example of what we think how they lived in the water column. So we think they were actually living vertically in the water column. Historically, people usually placed them in a horizontal position, but recent modeling studies by David Peterman, next month's speaker, has shown that they actually were probably oriented in a vertical fashion with their head oriented down. That's the most stable position. So it's also, I'll go and show you. So you can see this, and you see this with other planktivorous organisms actually living in the water column. So if you look at like the big fin squid, it lives in a vertical orientation and passively floats through the water column. So this is a big fin squid, has these incredibly long arms that it uses to collect small plankton and feed itself. You can also compare baculites to the oarfish. Now, the oarfish is most people don't know this, is they actually live in a vertical orientation and eat plankton swimming by, usually small zooplankton. The only difference compared to like a baculite is that the oarfish has its head pointing upwards rather than pointed downwards like a baculites. The big fin squid actually has its head pointed, its mouth pointed downwards like the baculites. So ecologically, baculites are probably very similar to the oarfish or big fin squid, except that they lived in very shallow water facies. So the oarfish and the big fin squid usually live in deeper water settings. Scaphitids. So Scaphitids are an interesting group. Here's a model of one made by David Peterman. Scaphitids are probably the second most common ammonoid that you'll find in North America. And in some species, especially as you approach the KPG boundary, they become the most abundant ammonite actually. So right before the mass extinction that killed all the dinosaurs and ammonites, these guys become the most abundant like group actually in North America and even other parts of the world, usually at temperate latitudes. We have a lot of data on this, and this is really thanks to Neil Lamon, who's a specialist on Scaphitids. What he's shown is that their stable isotope values are very similar to benthic organisms, which suggests that these guys were probably living close to the seafloor and living very probably within a few meters of the seafloor, and they weren't moving up or down. So throughout their entire life, they lived close to the seafloor. They like well-oxygenated facies with diverse benthic organisms. So that also kind of supports the interpretation that they were living near the sea bottom. In areas of the sea which had anoxic sea bottoms, these guys were not living. You could not find their shells in facies which are anoxic. Compared to like baculites, you'll find them in anoxic facies. So it's just they were living higher up in the water column. So what we think is that these guys were probably nectic planktivores, and the modeling studies by Peterman has actually shown that these guys likely had their heads, their mouth parts facing upwards. So they lived something like this, and they would actually catch prey swimming by or falling down from higher up. And so we also have their jaw structure. We think they were probably eating small, very small organisms like small gastropods, plankton gastropods, small crustaceans. And there's also preserved radula with these guys, or what they interpret as radula. And it's very similar to organisms that, well, cephalopods that eat plankton. So we think these guys were probably planktivores that were passively eating small little zooplankton. And their shells probably made them not very good for swimming. So they actually developed quite a few spines on them to probably prevent predation. So they have a lot of anti-predatory defenses, and those anti-predatory defenses seem to increase through the Cretaceous. So from their origins in Cenomanian or even Aptian, from the early Cretaceous all the way through to the end, they get progressively more spines on them. So they're probably not good swimmers actively moving. Most of these Ancylloceratids are really bad at swimming in general. The other groups that we commonly find in the geological record are the Nostocerids and the Diplomoceratids. And these are some of the most exciting heteromorphs. They're these kind of guys right here. This is a Nostoceratid called Didymoster cheyennense. So this is a model by David Peterman with a life reconstruction in it. We think these guys, based on how they float in the water column, probably were oriented like this. So with their head pointing upwards, and their jaw structure actually suggests they were probably eating pretty small prey. So probably small crustaceans, small shrimp, small things, just like the Scaphitids. And their stable isotope values suggest that they were living near the sea bottom. And it's also, that kind of data is also supported by like their facies they live in. So they don't like living in anoxic facies. They prefer living in well-oxygenated facies, usually in shallow marine environments, living close to the sea bottom. There's also the Diplomoceratids. These are the hairpinned ammonoids. These guys right here, we don't know much about their ecology. And I'm going to also make this point. We're not, so here's a Diplomoceratid right here. We don't know much about these guys. A recent modeling study suggests they probably oriented themselves something like this, kind of an incline above the seafloor. And stable isotope analysis suggests they probably live pretty close to the seafloor. And they probably shared the same habitats as Didymoceras. It's possible that these guys, these Diplomoceratids, are the males of the Didymoceras, but that really hasn't been well studied to this point. So that actually needs to be evaluated in the future. So we actually base that actually on their early coils. So if you notice right here, this is a picture of a Diplomoceratid. And when these guys, some of these guys, early coils are straight and some of them are coiled, like this one right here in this picture. When you have a coil early, like one that's coiled, you also have a Didymoceras that co-occurs with it that's also coiled. And when you have one that's straight, you also find a Didymoceras that has a straight early coil. So these guys might be actually male and female of the same Ammonite species. So we're not totally sure. This needs to actually be worked on. Most Ancylloceratids are ecologically similar to probably small forage fish. So fish that live in the water column and eat small zooplankton, and also modern squid that live in the water column. So ecologically, they take the same role. And this kind of summarizes what I interpret, like where these different ammonoids were living and what they were eating. So you have these planospiral, like Ammonitina, ammonites like Placenticeratidae, Sphenodiscids, living high up in the water column, eating small fish, gelatinous organisms. You have probably the Baculitids living in the mid-water column, eating like small zooplankton. And they're also possibly vertically migrating from the upper water column down to the lower water column, possibly in response to predators. We're not totally certain, that's speculation. And then like things like the Nostoceratids and the Scaphitids, these guys right here, they're probably living close to the sea bottom and eating zooplankton as well. So, and probably not migrate, they were never vertically migrating. So now that we've talked about like what ammonoids like were, where they lived and what they ate, let's talk about what was eating the ammonoids. So this is a key thing. And we have a lot of evidence of what was eating them based on evidence from predation. So the planospiral ammonoids, these Ammonitina, so the Placenticeratidae, things like Menuides and Sphenodiscids, they all show evidence of predation. There's only been there's actually been a lot of debate about this because people, they found these bite marks on these ammonoids. And there was a real classic study by Earl Kaufman, which from the 1960s of a Placenticeratidae with these distinct bite marks that match up pretty closely with the bite marks of a mosasaur. This is a mosasaur right here. People have debated if that's actually caused by a mosasaur or a limpet. There's increasing evidence and even unpublished evidence that suggests that they actually are not caused by limpets, but are caused by marine reptiles and probably fish. And so there's even like really amazing specimens where you see the bite marks on them and they're actually fractured up the shells, like the animal was still in there and then it was actually fractured. So like broken glass, but the animal kind of retained the shell around it. There's also evidence of like heel marks that look like they're punctured. A lot of this stuff has not been, this stuff that needs to be published on, but hasn't been in the last few, just recent discoveries. But I really do think like these guys were probably being preyed on by large predators. Another thing that kind of suggests they were eating by like large predators like fish and mosasaurs is you don't find their body chambers very often. So you have the septal parts remaining, but the part that's actually this where the animal's body fitted in is totally gone on most of these specimens. Just as being bitten by some large predator, and then the shell was being deposited close to where it was like living. Baculitids, they also show evidence of predation. It, again, not very well published on. So we do find little puncture marks on baculitids, actually on both sides, which is kind of interesting. Also, most baculitids, these guys right here, they're always broken up. And the question is, why are they always broken up? And some people have suggested, well, maybe they're breaking up just being like moved around in the sea bottom. And it doesn't always make sense that they're breaking up on this kind of soft, muddy sea bottom. What we actually think is that they are being preyed upon, and probably very frequently. And their shells were being, every time they were attacked, their shells would be deposited on the sea bottom. And you can even see, like, if you go through large collections of baculitids, you actually start seeing small marks that look like it was being punctured by some kind of fish or marine reptile. Scaphitids, these guys right here, they have really interesting evidence of predation. So if you look at them, they always have a puncture mark on the very rear part of the shell. So you see this puncture mark right here. And this puncture mark always corresponds to where the actual soft body of the ammonite attaches to the internal part of the shell. And what it kind of suggests, you even see like repair scars, and the repair scars usually occur when they occur like away from the puncture marks. So what we think actually is happening is that something like a fish or even a mantis shrimp is consuming these scaphitids. And so mantis shrimp, I don't know if anyone has ever seen a mantis shrimp, they punch everything. That's how they attack things. And so what it suggests is that these guys were living close to the sea bottom and were either getting bitten from behind and then, or were getting smacked by a mantis shrimp from behind. And you can even see like they have these prominent nodes on the rear part where they're getting preyed upon. So it's just they were actually becoming more spiny through time as an anti-predatory defense. So right in the area where they would be bitten. So you can see that corresponds perfectly where the spines are, where the bite marks are. So this is a chart that was made years ago by Earl Kaufman, and it shows the evidence of actually predation on baculitids and Placenticeratidae, Sphenodiscids, and also these ammonites called Nostoceratids. And you can see it actually increases the evidence of predation through the Cretaceous. And it seems to actually relate. You see it very frequently happening with Placenticerids and the Sphenodiscids in relation to like the evolution of larger and larger like mosasaurs. And the baculitids also start showing evidence of predation during the Campanian. You don't see it very often earlier on, but I don't think it's really been well studied. People have not really went through. This is some area that actually needs to be evaluated in greater like detail. And hopefully future research will actually start looking more evidence of predation in ammonoids. So if we were to look at what was probably eating ammonoids, you probably have like these Ammonitina, like the Placenticerids being by mosasaurs and large fish. The baculitids were probably being eaten by fish that were living in the midwater column. And then probably the Scaphitids and the Nostoceratids that were living near the bottom were being eaten by, well, you know, mantis shrimp and probably nectobenthic fish. So ecologically, ammonoids are very similar to modern forage fish. So they play probably a similar role to them. And some of the larger ammonoids, like the Placenticerids, were probably similar to larger predators in the ecosystem. So they played this kind of key role in between the zooplankton and like larger like vertebrates and stuff. And they probably were one step in the food chain. So if ammonoids were so, I mean, you find them abundantly in the record during the late Cretaceous. I mean, if you're going to find a fossil in any kind of marine rocks, it's likely going to be an ammonoid. Well, there's also these clams called inoceramids, which are more abundant. but if you're going to find any other fossil, it's going to be the ammonoids. They're very, very common. So what happened to these guys? So what killed them off? Why don't we have them here today? And why are a lot of these organisms like modern forage fish so abundant in the oceans? Well, if we go to the very end of the Cretaceous, you have a large impact event that likely blotted out the sky for a long period of time and probably killed all the plankton in the oceans. So all the photosynthesizers, and that probably had a trophic cascade like up through the different ecological tiers, which included the ammonoids. So ammonoids, as I mentioned before, preferred animals that ate zooplankton. So as you killed off all the zooplankton because they had no plankton, like the phytoplankton to eat, you wiped out their food source. So ammonoids were probably depleted because they lacked their food source. Also, ammonoids have very small eggs that probably, like colloids, that probably didn't have very long, like they hatched within 1 or 2 weeks. So they laid their eggs and then they hatched very shortly after. So they couldn't persist or live through a period of starvation. In contrast, like the nautiloids, so like the modern nautilus, they have very large yolks. When their eggs are laid on the seafloor, they actually will sit for 3 to, I think it's like 6 months on the seafloor. So if you have a period of starvation, these guys can live through it. Also, like nautilus are also scavengers. So if you kill off all the larger fish, larger marine reptiles, also the ammonoids, nautilus, which live today, would likely survive that because they were scavengers. They had a smorgasbord of different food resources to utilize. So ammonoids were disadvantaged because they were planktivores and they probably had very small young, which couldn't persist through any period of starvation. In contrast, the nautilus could persist for long periods of time, and even after they hatched, they probably had ample food supply because they were scavengers. So what did that do to the ecosystem? I kind of started mentioning how they have similar roles to a lot of fish. If you go to Cretaceous rocks, and this is actually kind of debated, Seibert and Norris looked at the abundance of fossil fish from a period before, during the Cretaceous, right after the Cretaceous, and then a little bit later on in the Cenozoic. And what you see is during the time when ammonites are abundant, fish teeth are actually very, very especially teeth of fish that would be like forage fish, are very rare in the geological record. But right after ammonoids go extinct, their teeth start becoming increasingly more abundant through time, which suggests that once you wiped out ammonites, you actually wiped out the incumbents at that kind of ecological level, and it allowed fish to come in and fill that similar ecological role. This work really needs to be studied more, and I think it's kind of an exciting idea that the loss of ammonoids kind of allowed modern osteichthyons, modern bony fish, to kind of start filling in those same ecological niches. So the loss of ammonoids probably led to the modern structure of the marine environment, so with a lot of modern forage fish. So here's my conclusions, but yeah, so just to summarize this, ammonoids lived in different parts of the water column and had different life strategies and ecological strategies. Most ammonites were probably relying off zooplankton and also probably gelatinous organisms. And they had an ecological niche that was similar to modern forage fish. And their extinction at the end of the Cretaceous possibly led to the diversification of modern bony fish in today's ocean. So I think that's it, but any questions at all?
Yes.
Okay.
First of all, a couple of, can you hear me?
Yes, I can hear you.
Okay. A couple of comments. First of all, Nautilus, the eggs will incubate for about 10 to 12 months.
Oh, 12 months. Okay, maybe I was wrong there about that. So,
well,
I say 6 months.
Again, this is, we're looking at wild, and it's difficult to figure it out. But if they incubated that long, the nautiloids could easily survive the Cretaceous. Second thing, the baculites. No ammonoid has chimeral deposits like nautiloids. So the baculites would have to be vertical unless they use liquid retention, which is possible. But
Yeah.
they had no cameral deposits to bring your buoyancy and center of gravity together. One other thing, this is my opinion, but the crenulated sutures revealing the crenulated septa, I look at that as not a strength, and this has been argued, not as
Yeah,
a strengthening,
Yeah,
but as ability to take and refill chambers with fluid, particularly when the fluid gets decoupled from the siphuncle. There's more surface area there that if the ammonite get damaged, they could refill a little bit quicker than Nautilus could. Last thing, there's an interesting paper that came out in 2021, Smith, Landman, and Creta, where they show that some hooks that they've been finding with Rhinoceros might actually be the hook pads on tentacles. So in their paper, they have a restoration where Rhinoceros has 8 arms and 2 tentacles with these hooks on. The hooks were way too big to be radula hooks, and they've actually found radulas for Rhinoceros, but that is a really interesting paper. Smith, Landman, Creta, 2021.
Oh yeah, I'm familiar. Yeah, I'm very familiar with that paper.
That is incredible. And of course, there's another person I'm going to mispronounce his name, Momenko. I believe that always has shown ammonoid restorations with 8 arms and 2 tentacles. I'm mispronouncing his name, I'm sure.
Did I meet you at the Burpee Museum one time?
Quite possibly.
I think I was there, and I think we talked actually.
That could be.
You're a big cephalopod collector, aren't you?
I dabble in nautiloids. Let's just say that. Let's just say that. I was also the person that wrote the amateur's perspective in PRI's American Paleontologist.
Oh yeah.
Way back in the day.
Yeah, that's cool.
So anyway, there you are.
Yeah.
Question
cool.
and question and a comment when you get time.
Oh, sure. Yeah. Are you finished, John?
I am finished.
Okay, thank you. Great points. So, Malcolm?
Question and a comment. Comment, Sphenodiscids have non-truncated venters and very compressed shells, which to me indicates fast swimming.
Yeah.
The second thing I question, with your Hoploscaphites tentacle reconstruction, was any of this derived from soft tissue recoveries?
They haven't found the soft tissues of Scaphitids yet. There are soft tissues from Jurassic ammonoids and also from like Cenomanian ammonoids, but I don't think they have found any actually for the Scaphitids, especially the like Campanian maastrichtium ones. I don't know of any actual studies. Now, I'm trying to remember if they have them for like Allocrioceras and Scyphinoceras. They have like the eyes. It's really debated about. There might be like arm structures for some of these guys, but
there's some excellent stuff from England, Jurassic stuff,
yeah.
Belemnites.
The Belemnites probably are different. So they have probably thicker tissue, which gets preserved more readily than these ammonoids. They probably have real thin tissues that also break down more readily. Like colloids have much more. They're likely to be preserved more often.
Well, thanks for giving that some time. Thank you.
Thanks for the question.
I have a question too.
Okay.
What would you think is the likely lifestyle of Nipponites? The really weird. I'm not sure if it's.
Oh, Nipponites, yeah, that weird. Oh, that guy is crazy. Let's see. Can you see my screen?
Yep.
Okay, let's pull up a picture of Nipponites. Because if people don't know what this is.
You might have to re-share your screen with the picture window.
Let me get a picture of it. Hold on. Can you not see that?
No,
not yet.
Stop your share and restart.
Let me get a picture of one of these guys. I'm just gonna copy a reconstruction of it from real quickly.
Josh, minimize your PowerPoint. Maybe that'll
Can't-
work.
Let's see here. Okay, hold on. It's not doing it. Hold on. Let's see. There we go. Can you see that?
If you say stop sharing and then you say share again and then you click on the picture, it'll work.
Let's go. I'm going to open my PowerPoint up and I'm going to just real quickly paste it in here. Let's look at, here's Nipponites.
Also,
There you go.
There we go. Here's a Nipponites right here. Can you see that? Right there. Do you see Nipponites right there?
Yeah.
Yep.
Okay, that's Nipponites. Yeah, this guy. That's one of the weirdest, it's probably the weirdest ammonite that ever lived. I mean, it has the most bizarre shape. They're found all in the Pacific. So they're most frequently found in Japan and Sakhalin, and also they've been described, I think, also from Washington or Oregon. I have to remember actually. There's the road cut out there that produces these.
Yeah.
But these guys are strange. And I think these guys are actually living in the water column, and probably, it's weird. No one has really studied the ecology of these guys. I think they live in the water column. They're probably some type of weird passive planktivore that floats around, but I'm not really sure. And this is something that really needs to be worked on in the future. So they're a weird, amazing ammonite. Why do they have that form? Oh.
That would make sense since they're essentially a ball, which would be very, very, it would be able to float very easily up into the plankton.
Yeah.
I mean, it's just, and yes, the mathematics of that sucker is really incredible because it's an asymmetrical, random coil. It's defies description.
Yeah. They're pretty widely distributed in the Pacific too. So that's another suggestion that they were living in the upper water column. But if they were all planktivores, I mean, I think they're shallow water ammonoids. So they didn't really get out into the deeper water facies.
Okay.
They're living in up in the water column in the shallow water facies.
Okay.
So does
Just
that
a
answer your
warning.
question?
oh yeah, that's great. Yeah, looks like the
Just-
chat has an email for further questions because time might be close, but
Yeah, we've got 4 minutes left, less than
Oh,
4
okay.
minutes.
really great talk.
Thank you.
So I have a question. The jaws that you find, how often do you find them? In other words, are they well preserved like shells? Are they a softer tissue? What's their biochemistry and how frequently do you find them, like per 10 ammonites?
Oh, okay. So the baculite jaws, so we've done an analysis on that. We actually gathered a collection from the northern Black Hills of South Dakota. And out of 3,000 baculites, we find one baculite with jaws. So for every 3,000 that you collect, you'll find one with jaws. Scaphitids, their jaws are much more frequently found. So we find those probably around every 1 out of 100. So, and that's for the like Campanium ostrictian ones. We've never found the jaws of like earlier Scaphitids. Placenticeratids, all those other ammonites, never really, barely have been never found. We don't even know how. There's probably been 1 or 2 specimens found total. That's something that actually needs. I suspect people have found them. They just didn't know what they were. They also look like clams a lot of times. Their chemistry, they're usually calcitic with an organic layer, and that the calcite has a tendency to shed off, and just the organic layer gets preserved. So when you find them, they're usually sticking out of the shell, and it's by happenstance. I think they're more frequently preserved in the shells, but we just can't see within the shells. You have to CAT scan the shells to find them, and most people don't have that kind of equipment to go through and actually start CAT scanning hundreds and hundreds of shells to locate them. So they might be more common than we realize. It's just that we're not finding them. So that's a big bias. You have to be lucky enough to find, like anytime you find a jaw, to find them in place. So they might be more common than 1 in 3,000. So
I guess that explains why I never found one.
yeah, I think we think that actually happens a lot with the soft parts of ammonoids, is that their soft parts could be preserved in the shell, but you have to like get some kind of like equipment like CAT scanning to actually get in there and look at the internal parts to identify those things.
Hey, we are down to 1 minute. Josh, I want to thank you. And I put my email address in the chat. If anybody wants to continue a conversation with Josh, feel free to email me and I will happily pass it on to Josh. And I
Yeah,
am,
just I'm always happy
Sorry,
to
Bob.
respond to emails or chat if you ever want to. If anyone wants to ask more questions, happy to jump on Zoom with you. So.
because Tim's question looks like more than a 1 minute answer about isotopes.
Oh.
Nice presentation, Josh.
Josh, thanks a lot.
Thanks for the questions and everybody. Thanks for coming to my talk.
Excellent, excellent.
Thanks.
Josh, I guess I'll see you in a couple of weeks.
I'll see you in a couple weeks, JP.